Okay, all right. I think we'll get started. So this room, it looks very formal. But so this whole lecture series will be very informal. Interrupt me anytime to ask questions. The recording of this talk, at least for the slides and my voice, and also the PDFs or the any format, the PowerPoints, whatever. Will be saved online as well, so you can, go back and download all the slides from all the talks. Usually they get online about a week or so after the lecture. So, this course has been going on for, I would say almost a decade, and it's sort of evolved into a format of 3 lectures a week, and usually by some expert at the NIH, talking about what they know best. And this one is no different than that, other than we tried to make it more not only didactic, but also sort of have them pose, open-ended sort of problems or open-ended sort of questions. And so as you can see, I don't know if you had a chance to look at the course schedule either in the email or past emails or Google Sheets, but it basically covers everything. Roughly at the beginning, I know you can't see this, I can even barely see it up here, but roughly at the beginning, the first several lectures will be like the basics of MRI, fMRI, the nuts and bolts of imaging, and then it will go into some of the more advanced processing methods of fMRI, and then it will then start to get more towards and also some interesting applications like real time and things like that. Then it'll go into more of the cutting edge. And just one small thing, some things might be rearranged as we go along, so keep on checking back with the Google Sheets to keep an updated list. I think it's pretty settled in, but I just made a change that didn't appear here a couple days ago, but it's mostly settled into this schedule. So then later on, it will go into other techniques. About mid-July, it will shift gears into PET and neuromodulation and MEG, and then move into some of the statistical processing, and finally some of the applications as well. And like I mentioned, I sort of instructed all the speakers to sort of keep an eye on some of the questions that are involved, and I guess that's it. So I will. So my lecture is going to be on sort of giving an overview on fMRI, and it will be sort of not only it will be a little bit of basics, a little bit of history, a little bit of sort of open-ended questions as well, and challenges that the field faces, maybe potential research directions. And also, it will sort of include some very cutting-edge stuff that I'll just touch upon briefly, because many of the other lectures, so it's sort of an introduction to all the fMRI lectures in some sense. At least it's an attempt at that without being too crazy and overwhelming. And lastly, it will be pretty much, so there might be some content that is very basic, too basic for some of you. Might be some content that's more advanced. Maybe some of you who've done fMRI have seen a lot of this before. But it's basically just to try to build a perspective, build a feel for the field in some sense. Okay, so I'm Peter Bandettini. I'm actually in charge of the functional MRI core facility. We have, within the core facility, we have a machine learning group and a data sharing group. I also have my section on functional imaging methods, in which I explore pulling as much information from the data, both physiologic and neuronal, and looking at contrast mechanisms. Okay, so just a quick overview. It's been 27 years since functional MRI began in 91. There's currently over about 4,000 papers published per year, over 52,000 in total. And it's unique in a sense that it's one of the only modalities that really sits at the interface of a huge range of disciplines. It's not invasive, fast. It has whole brain imaging. It allows whole brain and high resolution. This is very unique among all the human brain imaging techniques available. In particular, the non-invasiveness and the rapidity of it. It's had a lot of impact, at least in understanding how we understanding the brain itself, how it's organized, at least at the systems level. Many disorders have been studied. At least most of what we do here at the NIH is compare clinical populations to normal controls. And so we do group studies for the most part, but we're moving more towards individual assessments. So it's done a lot of that, yet there's no major clinical applications yet. There's maybe pre-surgical mapping a little bit on individual subjects, but the big problem is that for all of our neuropsychologic studies or all our studies on clinical populations, we might derive information, just general information about how brains are different, like their connectivity or their activity between clinical populations after we average it together. But that data might be useful for understanding things, but as far as useful clinically, it's almost useless, because what's really useful is be able to put a subject in the scanner and be able to say something about that subject, what population they belong to. So that's something to think about. So there's my course. So the organization of the course is this. I'll talk about how it all began, first challenges and opportunities back then, what's improved, what's not improved, current challenges, and where are we going, and finally the ultimate limits and applications. So how did it all begin? I always like to start with this slide, because this was a paper published in Science in 1990 by, most of these people were here at the NIH at the time, and they were leading the field. And what's interesting is that not only what it shows all kinds of types of functional MRI, metabolic imaging, magnetization transfer, diffusion, angiography, perfusion imaging with gadolinium, metabolic mapping, but it completely misses fMRI. And that's just to sort of give you an indication of how quickly fMRI came upon the scene. So fMRI was discovered about 1991. This was in 1990. There was no hint that fMRI was going to be discovered. So that's actually telling in a sense that you never know when something new comes along as far as learning to embrace it quickly. And it might be right around the corner. Who knows? So the first functional MRI work was not bold contrast, but it was done by Jack Beliveau at the MGH back in 1991, and he was using a blood volume tracking technique. It was an injection of gadolinium, a contrast agent that's a susceptibility contrast agent that, as it basically goes through the bloodstream, this is a time course of images, once it reaches the brain and it goes in, it causes a massive amount of signal dephasing. And I'll get into some of that. It basically causes, because it perturbs the magnetic field on a microscopic level, it causes all the spins to dephase. And they destructively add, and the signal goes down. And as it washes out, you can see the signal goes back up. Integrating under this curve on a voxel-wise basis, he was able to make a blood volume map that was pretty quantitative. And then he did it again during activation. And during activation, it's actually this curve right here. The arrival time is slightly faster, and the overall volume is slightly larger. So subtracting these two maps, he came up with this. And this ended up being the iconic sort of picture that has been, was on Science, November 1991 issue. That sort of inspired all of us. I was actually a graduate student at the time, and I saw this. Actually, I saw his work presented at ISRM. He won the Young Investigator Award back then. And it was really, really interesting. So what about bold? So bold contrast has sort of been worked on in some sense since a long time ago. Linus Pauling first characterized blood susceptibility with oxygenation, the unique change in its magnetic properties, and the ability to distort magnetic fields varies with oxygenation. He understood that. Keith Thilborn, who was at the time a spectroscopist, in 1982 characterized the fact that the blood T2, the relaxation rate, changes with oxygenation. But it's interesting. He didn't really pursue it as far as thinking that it could be used for looking at brain activation. In some sense, the spectroscopists and physicists and physiologists didn't really talk as much to the neuroscientists. So There wasn't this sort of like, oh, we can maybe use this to map brain function. It was more for physiology. Ogawa came along. He was working at Bell Labs at the time, and he actually had coined the term bold, and he noticed that T2 and T2 star, and T2 star was a lot more, and you'll get in later lectures, we'll talk about the difference between these two. T2 star is actually much more sensitive to magnetic field homogeneities, inhomogeneities. T2 is sensitive to more microscopic inhomogeneities at the level of the capillary. So he found that they were changed by changing the oxygenation in rat brains by modulating their oxidation level. Bob Turner showed the same thing, and he did this by cutting off the oxygen to cats and looking at the whole brain get dark and back light again. In the process, he was actually looking for diffusion changes, and he kept on seeing these T2 star changes and shifted gears and moved towards in that direction. Okay, and the idea here is that you have, with red blood cells, with vessels containing red blood cells, essentially you can look at the body or any object that has the same susceptibility as the surrounding area as being matching it's either paramagnetic or diamagnetic. Everything is slightly diamagnetic, most biologic tissue. So if you have that in the magnetic fields, it doesn't distort anything. But if you have deoxygenated blood, it's slightly paramagnetic relative to the rest of tissue. In fact, deoxygenated blood is still diamagnetic, but it's less diamagnetic than the rest of tissue. That distorts the fields, that causes the spin dephasing, causing everything to get dark. And this is kind of what it looks like. This is Ogawa's early work, a rat brain showing 100% oxygen, not much, no lines that are dark or anything like that. If you give it 20% oxygen, like room air, the metabolic rate of the rat will pull oxygen further from the blood, and you have these dark lines forming. And this is all due to deoxyhemoglobin. And he did the same sort of experiments in test tubes, 100% oxygenated blood, 0%, and you get this massive penumbra. Okay, and the first experiments that were actually shown, I think, publicly was at the 1991 ISMRM meeting. The person who was in charge of the NGH NMR Center, Tom Brady, was giving a plenary, and he just threw this up and said, oh yeah, so we're also doing this thing called, MRI, and we're looking at brain function, and you can see these movies, and the brain lights up when a person gets visual stimulation. And I was actually sitting in the audience at the time, and I think everyone's jaw kind of dropped. And they looked at this, and I was actually looking for a thesis project at the time, and I saw this, and I realized this is what I want to do. And I was lucky enough to be working with a colleague of mine, Eric Wong. This is us back in 1991 building a head grading coil. And the reason why we were building a head grading coil was because the scanners at the time weren't equipped to do what's called echoplanar imaging. It's a very rapid imaging technique for where you drive the gradients. All of you who have done fMRI, you hear the ping, 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 ping. That's echoplanar. And it makes that high frequency pitch because the gradients are oscillating so rapidly. And so we had to make this low inductance coil so we didn't blow up the gradient amplifiers when we did echoplanar imaging. And we were pursuing it for a different purpose at first. Eric wanted to do perfusion imaging in the brain. He needed echoplanar imaging. But we were ready to hit the ground running once ISMRM happened to do functional MRI on our scanner. And this is how the gradient coil looks. It was just simply placed on the scanner table, which was potentially kind of dangerous, but we didn't have any IRB. We didn't have any, back then, we didn't have any sort of safety committee or anything like that. So we were working at like 2 in the morning, and I was the guinea pig for a lot of the experiments. This is me in various versions of our scanner. And it worked pretty well. So this is our first experiment. I basically pulled out an anatomy book and then looked at a coronal section and said, okay, where do I pick? Because we only could pick one slice at a time. And so I said, okay, let's just pick a big slice, 2.5 centimeters thick, just getting a chunk of the motor cortex right here. And that's what we did. It was TR was 2 seconds, TE was 50 milliseconds. We knew to have a longer TE to optimize it to the T2 star changes. That's at least one thing we gathered from the ISMRM meeting before. And this is the movie. This is a movie, an early movie from an axial slice of my brain. tapping my left fingers, tapping my right fingers. And so it's displayed sort of in a time series of images in which the average, or the first image, where there's no activation, either of left or right, is subtracted from all the following ones. And so you can see clearly the left and right motor cortex lighting up. You can also see some early hints of resting state fluctuations. Some of it's breathing, some of it's heart rate, and some of it's real time course activation that wasn't going to be really understood until, or discovered until 1995. But it's all there. So here's our, we had our first, lucky enough, we were lucky to have one of the first papers. Ken Kwong, though, I think was really the pioneer of this. He was the one who did really some of the first, he really conceived of some of the first experiments. This was the initial data that came from the MGH lab that Tom Brady showed. He actually took it one step further. He actually developed a T1 sequence that's sensitive to inflow or perfusion in the brain that also showed brain activation changes with a slightly different time course than BOLD. This is BOLD, and this is perfusion. And he also even took one step further in his PNAS article of comparing it to activation as it was measured with PET imaging, positron emission tomography with, I think it was 015, just flow imaging. So, and then the Minnesota group was also very active at the time because they were collaborating with Seiji Ogawa. And they waited, they could have been even earlier, but they waited until their 4 Tesla scanner was up and running because they thought that they couldn't get it running at 1.5 Tesla, even though both of those first experiments were at 1.5 Tesla, but they both had echoplanar imaging. They were using multi-shot imaging, but they did a little bit higher resolution at 4 Tesla. And one thing that they advanced was that it truly was a susceptibility mechanism. And the way they did that is showed that at short TEs, you don't get much signal change. At longer TEs, you get a larger signal change. So because it's TE dependent, that shows it's related to T2 star changes. And you'll probably hear more about those mechanisms later. So that's where the field was. And since then, it's done a lot of advancements. You have, and I like to put it in this framework, you have technology, methodology, interpretation of the signal, and applications. And they're all sort of working together, driving the field. And in my opinion, you can always identify the best work in the field because it's right about here. It's right at this interface between the best use of the technology, the best methodology, the most insightful and accurate interpretation of the signal, and it involves some interesting question or application. So, but really they've been driving each other. With technology, you get better signal to noise and better speed. Methodology makes the most of that. It optimizes the applications. And interpretation of the signal, we're still working on understanding things like fluctuations dynamics, and you're pulling information from there. So these 4 sort of pillars have been driving the field. Okay, so back then, what were the challenges? I put down 3. First, prove that it means something, characterize it, and then improve it. So back when I was writing that paper, I was trying to scour the literature as to anything that could suggest that a signal would go up, that oxygenation goes up. Because you would think that with activation, in fact, even Ogawa mentioned in one of his earlier papers that, hey, this susceptibility effect is interesting. Maybe with brain activation, the signal might go down because an increase in metabolic rate. He didn't consider at the time, I mean, it was very insightful, but at the same time, he wasn't talking to the physiologists who knew from this paper back in '86, this is Peter Fox and Mark Rakel's paper, that showed that, in fact, during activation, oxygenation goes up. So the oxygen extraction fraction goes down as they measured, and therefore the blood PO2 must go up in this case. This actually caused somewhat of a controversy at the time because they called it a decoupling. It's probably still a coupling, but it's just within a different linear regime. But nevertheless, that was sort of the paradoxical change. Everything with brain activation, the oxygenation actually gets higher. And this is the whole cascade of events that happen. Tissue activation, vasodilatation, causing cerebral blood flow and volume increases. This causes oxygen delivery to exceed the metabolic need. We don't still completely understand this. We have a good handle on it, but we haven't fully understood why this happens and why it happens so incredibly consistently everywhere in the brain, in every subject. Unless you have some real problems with your vasculature, it happens completely consistently. This causes a increased oxidation, and then it's a lucky thing in nature that deoxyhemoglobin has its magnetic properties. It's also fortunate that this is detectable then within the range of detectability as a change in T2 and T2 star, and that you see the signal increase. So what else did people do? Well, the first thing they did was try to parametrically manipulate the activation. So early studies were looking at motor cortex with different finger tapping rate, and they showed, oh, look, it goes up in proportion to finger tapping rate. I mean, this is interesting published study, but it's actually suspiciously linear. But I'm not going to question it. I'm actually a co-author on it, but I always wonder. I ask Steve Ray about that every once. So he's like, it just worked perfectly. It's great. And this is auditory cortex, in which the syllable rate is modulated, and so you also see sort of this monotonic response. And then a really important paper came along by Nikos Logothetis in 2001 in which he had implanted electrodes. And here he varied the contrast of a checkerboard and simultaneously looked at multi-unit activity and local field potentials with electrodes while he's simultaneously looking at bold while modifying the contrast in the flickering checkerboard. And he found that these are the basic measures, and he found that the relationship is quite linear within a certain area. He doesn't really talk much about this crazy nonlinearity that has to happen to actually have it go through zero. But which is actually very suggestive, because there's a lot of studies that suggest that with very weak activation, with very weak or very brief activation, you get more bold than you would expect. And this sort of predicts it, even though he doesn't talk about it in his paper. So it's nonlinear with weak activation or short activation. It becomes very linear proportionality to local field potentials within a certain regime, and it stays that way. Okay, what about the dynamics? So this is basically a block design. It takes about 9 seconds to plateau in the on state, a little bit longer to reach a baseline. Oftentimes you see some sort of a transient at the top here, you see a post-undershoot, or you see a pre-undershoot. You hardly ever see a pre-undershoot. That's still controversial. Most often you see a post-undershoot and an overshoot like that. With event-related, you can go incredibly short durations and still elicit a clear event-related response that typically peaks about 4 to 5 seconds after the stimulus starts. So how hard can you drive the system as if you're looking at an oscillating, its filter properties? So this is turning on and off the brain at various on-off frequencies. This is 20 seconds on, 20 seconds off, all the way down to 3 seconds on and 3 seconds off. And at least in this study, in the motor cortex, we found that it was driven it was actually a little bit longer to return to baseline, therefore it kind of gets saturated in the on state. But it represents exactly almost a sine wave at about 6 seconds on, 6 seconds off. And this is sort of the filter properties. There's an elbow right here at about a 12-second on-off cycle. But this is taken even further, even recently. So there's so many things that were sort of touched upon in the past that are sort of revisited with better processing, with better experiments, higher sensitivity, that even more insights obtained. This is some really nice work by John Polomini's group. This is Laura Lewis, who showed that if you even drive the signal, this is with a checkerboard at up to 0.5 hertz, this is showing up to 0.5 hertz, you still get a detectable oscillation. So now you're getting almost into the neuronal firing regimes. They even pushed it as far as 0.75 hertz. And what's interesting about this paper in PNAS was that it's not only like, oh, look, we can detect this, but we can detect this and it makes no sense at all because, once again, it shows that there's these nonlinearities in the bold response. A linear system would never predict that you could detect this occurring. So there's clearly some nonlinearities in the hemodynamic response signal that allows you to detect these very rapidly driven states. So this is the first event related, just for the sake of history. This is about, I think, the third paper published by Andrew Blameyer at Yale. He was working with Schulman, and they were just doing 3 seconds on with a visual stimulus. They showed an event-related response. They didn't call it event-related, so they tend not to get credit. Sometimes you have to label something to get credit for something. And once again, this is another example of, with event-related, how the system behaves nonlinearly. So this is some work by Rasmus Byrne showing that what you would predict in red from a linear system, and this is our various stimulus durations, and what you actually get in the bold response in your data. So it's extremely hyperlinear at the very low stimuli. And it turns out this has a spatial dependence as well. So we not only looked at the nonlinearity, but we mapped it. And we found that, at least in our data, that around the periphery, it behaves extremely nonlinear, much more nonlinear than around the foveal areas, up to about 2 seconds. And then it all starts to become linear again. And what's interesting is that, once again, Logothetis, with his great paper, he doesn't really mention this nonlinearity, that all the neural nonlinearities that occur, I mean, there's some massive amounts of, whenever you have looking at a spike train or local field potentials, there's a massive transient at the very beginning. And he doesn't really talk about this. He just talks about mostly the relationship between the overall signal, neural activity, and the hemodynamic response. So, and now, once again, this is something that has been revisited recently. And this is some work by Kalanick Grohl-Spector's group showing that these nonlinearities, we stopped there and saying, oh, it's around the periphery. And then we said, go figure, and we went on to other things. But then now, recently, there's a PNAS paper showing that there's extremely different maps for transient versus sustained contributions. And she models this to explain maybe that these transient contributions are doing computations that are completely different than the other parts of the visual cortex. So it's a really interesting paper in which she doesn't just say, oh, it's hemodynamic or whatever. She actually models it very carefully and tries to attribute functions to these specific areas that show this larger transient response, that they're doing some adaptation computation that's unique in some sense. So there's really in-depth neuroscience going on. Okay, so other aspects of the dynamics are how much temporal resolution can you get? So we can see that you can get very brief stimuli. You can collect very rapidly. You can collect as low as a TR of 100 milliseconds if you wanted to. 400 milliseconds for whole brain now. But it turns out that what you're really limited by is the hemodynamic response. And so it turns out that this is an early work which we had a hemodynamic response here, and we did a poor man's sort of latency map by shifting this right and left until we maximize the correlation with the response. And this is just bilateral finger tapping. And what we find is that the latency spread is plus or minus 2 seconds in the brain. And that just doesn't occur neuronally typically, especially within the motor cortex. And it turns out the magnitude is also varies. But if you look very carefully at these patterns, the magnitude matches the latency. And the longest latency in particular seems like over this dot here. And that's over a dot right here with the largest magnitude, with creating a venogram that's right over a vessel. And so you might get this distribution of latencies, but really the long latencies are typically over the largest veins, largest draining veins. And so that's what they might be downstream collecting oxygenated blood that takes about a second or two to get to them. So that's a real problem that we're still grappling with, because you have to first do either a vessel mask or characterize the latency in some ways to pull more information out. All right, what about spatially? So this is some early work on ocular dominance columns by Ravi Menon and Kang Cheng that seem to map very closely with Frostig's work in optical imaging. And this was sort of very much of a breakthrough at the time. This is way back in 97, 2001. And there's also this work, which has since then become even, people at the NIH here are sort of re-looking at this and realizing that, so with this group, this is Essa Yaqub at Minnesota, 2008, it was published 10 years ago. Nobody's really repeated this yet. This is actually looking at orientation columns. And that really pushed the field towards, increased field strength and other techniques. It was a very hard experiment to do. However, and this is also ocular dominance columns are here. So the orientation columns are much smaller than the ocular dominance columns. Turns out you have to be very careful in terms of how you process the data. So while it's not clear whether this is real or not, it's compelling, but at the same time, it's potentially easy to create these maps, believe it or not, by not being careful about how you interpret your processing. So it's, anyway, that's just basically, that shows the very bleeding edge in terms of columns, in terms of what the resolution is. And it's still controversial right there. Okay, so the first opportunities. What could be the new contrast? What could be the new paradigms? What are the new processing methods, new applications? So as far as fMRI contrast is concerned, there's a whole slew of new contrasts that came about. Of course, you had invasive volume imaging, you had bold contrast, perfusion contrast came out at the same time. Wasn't as successful because it was less sensitive and it took longer in terms of temporal efficiency. Then by looking at the ratio of perfusion and bold, you can actually get measures of CMRO2. That came out in about 1998. So metabolic measures. Vaso, this technique that is looking at just blood volume changes, but non-invasively. Sort of like an arterial spin labeling, but basically putting in an inversion pulse and allowing the spins to relax. And basically, if you would do an inversion pulse, all the magnetization is negative, and then it goes through a null point. And it has different relaxation rates. Blood has a different relaxation rate than tissue. If the blood goes through the null point, there's some signal with the tissue. And then they image again. And so you get everything is dark where there's blood. And the idea that if there's more blood, more is dark. So that's very sensitive to blood volume changes. Then other experimental techniques are neural currents. There's theoretically possible to detect just with the same, the dipoles that produce the MEG and EEG signal at the source. They produce a magnetic field that would cause spin dephasing or phase shifts, and that's theoretically detectable as well. So far, no one has done it. I think it's about an order of magnitude smaller, and all the images are dominated by bold, so it's hard. Other people have come up with looking at diffusion coefficient changes, like cell swelling. Denis Lebihan, a prominent person in the field, has sort of banked his entire career on this, on claiming that if you have a B factor diffusion weighting that's high enough, and if you average enough, you can see cell swelling with activation. As can occur, but whether it's detectable, that's another question. Temperature changes, that's been measured with activation. There's a recently work that came out in last year's ISMRM meeting that claims that elastography changes occur. The brain gets, at least at the meeting, they claim the brain got more stiff, and now they're claiming the brain gets less stiff. So I don't know. It's all up for grabs right now. Okay, so now there was also a massive explosion of types of paradigms. So we started with block design, kind of taking from the PET literature, 20 seconds off, 20 seconds on. Very easy to process. It's actually nice to use paradigms like this because you're constantly fighting against signal drift. And so if you have a block design like this, if the signal is drifting, then you can actually pull this high frequency out from the signal drift. So if you have a very low frequency, like if you just did 10 minutes off and 10 minutes on, you would be contaminated by the drift effect. And so that's why we do go on, off, on, off. Frequency encoding. This is sort of an esoteric technique, but we did it. Basically inputting activation at different frequencies and pulling out by simple Fourier analysis. Phase encoding, this has been used extensively in retinotopy, tonotopy, somatotopy, where you can actually change the type of the characteristics of the stimuli in a continuous fashion, and then map as they change what changes in the brain. So it's a very powerful technique as far as that's concerned, because you're always doing something. So there's event-related designs that are either jittered or constant. We now use jittered event-related designs, assuming a linear system, so we can deconvolve the neural input or the hemodynamic response from the neural activity. And that's a pretty good assumption for the ranges that we do it in. fMRI adaptation, it was developed by Kalanick-Grylls-Spector as well, in which you modulate the similarity of the stimuli or the task in a rapid succession. So if neurons get habituated, they cause less activation. So this is a way you can tune very carefully neural populations within a voxel. If the stimulus is a little bit different, then you'll get a little bit less habituation neuronally, because different neurons are firing in that same voxel to the activation. So it's a way of sort of pulling out what populations of neural pools are in a voxel based on the fMRI signal adaptation. And then we design stimuli to be orthogonal because that was statistically most powerful. You can actually orthogonalize mathematically anything, but you throw away statistical power. So it's always nice to orthogonalize your block design up front as much as possible. And then free behavior design. Sorry, this is an example of free behavior design is sort of allowing the subject to behave naturally in the scanner. I'll give you an example of that. And that's, I think, where the field is going in that regard. This is just an early example by Steve Engel at Stanford of retinopathy, in which he had various rings separated by blank areas that are constantly expanding outward. And he was able to create these wonderful sort of traveling waves as the rings expanded outward very slowly. It was like one cycle every minute of expansion. But you actually could create these nice traveling waves that then you could do retinopathy with. This is an example from work by Ted Dio, measuring either retinopathy, and this is coded with where the stimulus was and what the activation was, and so you can see nice peripheral organization. And then you can encode for wedges that rotate. And the key to the rotation is that different visual areas of V1, V2, and V3 have opposite directions in which this traveling wave travels. And so you can actually then discern boundaries of the visual areas by where the activation either runs into each other or runs apart from each other. So that way they're able to map out early visual areas very, very easily. Okay, then event-related design was finally applied to cognitive activation. This is some work by Randy Buckner. It was also done by Greg McCarthy at Yale as well, in which this case, it was just a word stem completion task, focusing in on the left anterior prefrontal gyrus. With a word stem, you could elicit a nice hemodynamic response. So that was the first cognitive application of event-related right there. And then the field took off, in which Mark Burrock and others sort of realized it behaves as a linear system, and so you could do these various things like jittering, randomizing the sequence to actually get more stimuli in and then deconvolving the hemodynamic response very effectively. So that's sort of was an important driver for the field. And so this is one of the first examples of free behavior design. This is some work by Eleanor McGuire's lab, in which she had subjects just driving through a simulated London right here. And they did various things, and they categorized what they had to do here, and then they just simply continuously collected the data and then binned it based on what they were doing. So all these are various bins that correspond to these color codes. And depending on what they were doing, then the bins were averaged together, and they made these beautiful maps that looked very different from each other depending on what they were doing. So this is very powerful because the subject was freely behaving, and they were doing very complicated things, but you were keeping track of the complicated things that they were doing, and you allowed the paradigm to just determine how you binned your data. Okay, so the big revolution came with Bharat Biswal back in 1995. He was the first person to do resting state fMRI. And what's interesting is that he couldn't have done it without an early version of AFNI. AFNI is a processing platform that Bob Cox created. But before Bob Cox started working on it, it was a very basic tool that Bob, I think, early on developed. and also in combination with Andrei Jasmanowicz, who was a physicist at their center, and basically allows you to pick. It was very efficient because most studies, you would have to create a reference function to pull out the information of the data. This one said, okay, you don't have to create a reference function, just pick some area from the brain and use that as a reference function. So it was a very quick and dirty way of assessing your data, typically when it had activation. But Barat was the first one to actually say, let's just see what happens if I apply this reference function to resting state data. And so this is what happens. So here's task activation. He found left to right motor cortex that were shown to be in the right places. And then in this case, the subject was just lying in the scanner doing absolutely nothing. And Barat just took a voxel from this part of the brain, used it as a reference function. It looked like nothing. I mean, it looks like noise, pretty much. But lo and behold, it's the other side correlated highly with it, about a correlation of 0.3, when typically with activation you get 0.5 to 0.8. But still, it was correlation. And it clearly showed the same or similar pattern as activation. And he published this up, and resting state fMRI began in 1995. What's interesting, though, is that back in 1995, when he did it, everyone was just starting doing their activation studies. And most scanners weren't this sensitive, and processing methods weren't really keeping up. Believe it or not, we didn't really have an ability in other packages to pick a voxel as a reference function. I know it's very easy, MATLAB or whatever now, but back then it was something odd. And so everyone ignored resting state fMRI for a decade. So nobody cared. There were maybe 1 or 2 papers trickling in over the years. It finally took off about 2005. So, okay, so when it did take off, other techniques that are much more efficient than picking a seed voxel picked up. This was work by Steve Smith's group back in 2009, doing one of the first analysis using ICA, pulling out these independent, spatially independent component networks. And comparing that to task, and you see a lot of spatial similarity. Okay, and so then the field advanced. Here's looking at just once again, there are uses for seed voxels. There's limitations to ICA, various networks being pulled out with various seeds. And this is some really recent work by Arcaro, which was really dramatic, showing that you could do retinotopy with seed-based resting state, in which you move systematically the seed to more peripheral regions, and you get a clear retinotopic organization. So that was nice. You could do the same thing with mototopy. So here's a movie in which you move the seed along the motor cortex, and you see this area of activation, on both sides increase and then decrease and so on. And even more interesting, this is a nice dramatic movie by Randy Bruckner showing you can do this on the whole brain. So this is just something where he had super average resting state data. I think it was like 1,000 subjects concatenated, and showing that if you move this thing around, this seed voxel, all these other areas sort of show increases in correlation as you just randomly move that seed voxel around, which is just fascinating to look at. AFNI has a function that does this as well. So it's Instacore, basically, it's called right now. All right, so from there, a lot of groups have developed parcellation schemes based on resting state connectivity. And this is using hierarchical clustering. This happens to be multi-echo data, which we have our own way of going about doing it. This is work by Prantik Kundu, who was a graduate student in my group back at the time of doing this. Is about like 5 years ago. And here he showed that there's clear, it looks basically like a Brodmann's map now. So now you can actually, from 7 to 10 minutes of resting state data, you can create something that looks like a Brodmann's map in individual subjects. This happens to be an average of 7 subjects, but we're at the point now where we're beginning to parcel with individual subjects. So, and this brings to mind actually one interesting point, is that coming up with universal parcellation schemes for subjects is difficult. So each subject has their own parcellation, and maybe the distribution of these parcellations is a biomarker in itself. But if you want to start averaging together subjects' connectivities, it's efficient to first parcel, But then you want to make sure that you get similar parcels across subjects. And that's tricky. And so there's work on that being done right now as far as that's concerned. Okay, so resting state took off. Now it's kind of gone through the roof. This is up to 2014. It's probably on equal grounds with fMRI itself. So other processing methods, Jim Haxby, but when he was here at the NIH in 2001, was studying face areas in the brain. So he actually was one of the first people to sort of find face areas. And he said, oh, there's a blob that shows an area of activation with faces. And it's different than houses, for instance. And so, but he tried to expand this to find other blobs. He did chairs and scissors and shoes and cats and things like that, and actually realized it was no longer a blob. So this happens to be like one typical activation with a chair versus in here happens to be with a shoe. It's not really, it never really becomes clear. It sort of starts to look to the eye like more of a salt and pepper pattern that other researchers might look at that and say, oh, the experiment didn't work. So Jim did something really good. He sort of rediscovered multivariate pattern assessment in the context of fMRI. So he said, okay, let's just take this pattern, whatever it is. It doesn't look like much, but let's take this activation pattern and see and split the data in half and see how it correlates with the other half of the data from that similar stimuli, and then see how this pattern correlates with other stimuli, other data from other stimuli. And he found that there was a high degree of correlation with the same types of stimuli and lower with other stimuli. So he was able to say, oh, there's still information in this data, even though your eye can't see blobs of activation. Instead, it's sort of distributed at high-frequency patterns. And that sort of started the field of multivariate pattern assessment. Nico Kraigasorti, who was a postdoc in my group, essentially did an experiment where he created a large matrix of different stimuli and simply looked at how the patterns matched. So he took it to its extreme and used all these stimuli, everything from faces or animate objects and whatnot, and found and put it in a matrix correlation. He found that that they clustered. There are similar patterns for humans versus or human versus not human, animate versus inanimate, and so on. In human area, in visual area IT. In early visual cortex, there's no such pattern. If you were to, and I haven't really seen this study, but if you were to actually probably reorganize how you look at these patterns instead of humans versus not humans or animate versus inanimate, you just organize them based on spatial features or low-level features, then maybe V1 might show patterns as well. So it's a way of pulling out very efficiently, casting a broad net for information from the data. So since then, the new applications have picked up. First couple of years, people were just doing proof of concept experiments, but now, by about 1998, they really believe the signal. The NIH granting agencies believed the signal, and people were starting to do all kinds of things, everything from basic cognition to motion, higher order cognition, and so on. And this is just a summary, if you can look at later, of the way I sort of categorized all the basic developments in the field from technology to methodology, interpretation, applications, up to 2003. Now it's sort of completely ballooned into all kinds of things. And if you're curious, there's actually a special issue that came out in 2012, I believe, on the 20 years of just all the methods advancements. What's nice about this is that the papers are sort of first-person sort of stories on what that author did and what their contribution was. And so it's a really fun read. Okay, now this will go faster. What's improved? All right, image quality, temporal stability, sensitivity, resolution, and speed, implementation, standardization, processing, display. There's many different processing methods available now. It's just a lot of variety. Artifacts are better understood, more fully removed. Underlying neural correlates are better understood. Resting state has exploded. Large pooled data sets now have catalyzed meta-analysis, transparency, new methods, biomarker discovery. Individual assessment in fMRI is growing. Standards, the basic resolution used has gone up to about 2 millimeters as opposed to 3.5. There's a lot of efficient sort of streamlined integration of multiple modalities. More people are using it, better questions, and more high field scanners. So this is a map created by Laurentius Huber. It's actually online, and it's constantly updated of all the 7Ts. There's over 60 of them, 60 throughout the world. Okay, what's not improved? This is something to be focused on here because these are the areas where research might be useful. We're still struggling with subject motion and breathing. So even if you nail the subject's head to the scanner completely, you will still have the head pulsating with every heartbeat. With every breath, what happens is that the B field shifts, causing that spills into the brain that creates an apparent motion because you have a slightly different B field in the brain. So these are all problems that need to be dealt with. Temporal signal noise is still limited by physiologic fluctuations. We're still struggling with understanding the hemodynamic response function and how it varies over space and how it varies across populations. It's still, if we better understand that, we might be able to pull out more temporal information, more causality information, and so on. The spatial resolution of fMRI has reached pretty much a theoretical limit because at least the most refined control of hemodynamics in the brain has been on the level of columns and layers. So they haven't found that the brain controls hemodynamics at any smaller resolution. So if you have a technique that is based on hemodynamics, we're there. We're at the limit, which is okay. There's a lot of work that can be done. fMRI is still not used clinically. The effect size to noise, at least in understanding multiple subjects, is low. Vendors still don't care about fMRI that much at all. We're actually lucky because MRI is so successful that we're sort of riding on the back of all the success that MRI has had with other applications. If MRI wasn't a clinically useful, if MRI in general wasn't a clinically useful technique, our scanners would be 10 times more expensive. And there would be maybe 1/10 the number of people doing the research on fMRI because of that. So we're lucky for vendors, but unfortunately we can't get their attention to pay more attention to fMRI. Standards are still loud and confining. MRI is still very expensive, even with all that success. We're still using 2.5 millimeter voxels for most studies, which we can do better. And this is an important point that I always like to harp on a little bit, that even though resting state functional connectivity works so well, we actually don't understand what it's for. I mean, why does the brain show this connectivity at these low frequencies, and why do oscillations, so there's many other ways of changing the correlation of two time series other than changing the connectivity of those regions. You can change the amplitude of one, you can change the noise, you can change the latency, all that changes correlation, but it might have nothing to do with connectivity. So we really need to dig down deeper in understanding the relationship between our correlation of our signal and our connectivity. We don't understand the source of resting state fluctuations, as I kind of implied. We don't know what the biologic purpose that it serves, or if it does serve a purpose. Shimming is still an issue. Vendors feel the shimming is good enough for the most part, but if we're going to 7 Tesla, we're looking at base of the brain structures, we really need better shimming. It's theoretically possible, but we're actually at a limit in how fast we can image not because of any technology, but because we can't switch the gradients any faster, because that will cause peripheral nerve stimulation in the body. So we're up against that limit. And we have many large databases, but we really need to have them better curated. We have to have the data more standardized in these databases. Okay, I'm going to quickly go through this. The few current challenges: completely remove fluctuations, create a pipeline for a robust individual assessment, high-resolution mapping that's impervious to vascular variations. So this is just a quick plot showing you the temporal signal to noise of the data versus the image signal to noise. So this is actually the signal to noise determined by the RF coil. And this is the temporal signal to noise. And it turns out that if you get to a high enough image signal to noise, if you're looking at the temporal signal noise of gray matter, it plateaus. Plateaus at about 150 to 1. And so you could have a signal to noise in your image. If you measure it by taking like an ROI in the gray matter, in the brain versus outside, that could be 10,000 to 1. You could have the most clean image in the world. Your temporal signal noise will be 150 to 1 because we haven't figured out how to get rid of physiologic noise. So if we do get rid of physiologic noise, then our limit is only determined by the RF coil. Our sensitivity could go up by an order of magnitude. We just need to model it better. It's a really hard problem. Okay, another problem is coming up with the universal parcellation scheme. Glasser has made, I think, one of the best progress on that in their Nature paper. I'm not going to get into it for the sake of time. Another issue is individual assessment. And there have been some groups that have, a lot of groups that have started working in these directions. So the idea here is that, this is just one example, where just tons of examples in the literature, looking at fluid intelligence. Some groups have claimed that there's biomarkers of cortical thickness and functional connectivity that seem to show a nice relationship between fluid intelligence and these measures. This scatter around this line is good. It's great for group studies. It's completely almost useless for individual assessment. There's still too much variation around this line to actually get at individual assessment. So, and there's other methods as well that show this as well. So I'm going to skip this slide because this is more of a reiteration of the things that have the best potential. So depression, understanding traumatic brain injury, stroke, Alzheimer's disease, all these are relatively not developed. They're not very mature as far as a functional imaging standpoint is concerned. And the size of the circle is the amount of impact that you could have by doing something about them. So there's a lot of opportunity here as far as pursuing these sort of diseases with neuroimaging. I'm going to completely skip this work because Renzo Huber is going to talk about this at much more detail. And I will come up with I'm going to skip this because this is going to be talked about by Javier Gonzalez-Castillo. I just want to touch on this one slide here because he may not talk about it because he's talking about multi-echo. Is that even what we think of as activation should be put into question. This is a work, and this is something that, these are things that directions the field might go. This is work where Javier Gonzalez-Castillo, who's a staff scientist in my group, averages subject for 9 hours, doing one very simple task, staring at a fixation point, and then when a checkerboard came on, it had either a number or a letter in it or nothing, and they had to press the button if they saw the number or letter. Very motor, visual, very simple. And what he did was, of course, he found motor cortex activation, he found visual cortex activation, but when he averaged up everything, and then loosened up what models we were using to pull out the information from the data. It wasn't a canonical hemodynamic response. It was the hemodynamic response, but it wasn't the canonical timing that you would expect, like a block design like this plot right here. It turns out the brain is doing all kinds of stuff that is time locked, that does not resemble that block design. And if you time lock it and you average together and then you just use simple clustering, this is hierarchical clustering to group those patterns together. The whole brain, all the gray matter essentially is active. It's all active. It's all doing something, either responding transiently at the beginning, transiently at the end, negative signal changes, then positive signal changes, and so on. So if we have more sensitivity, all this transient, interesting activity could come out. So it's all there, and it all is real brain activation. It's just a matter of pulling it out. Okay, and then there's, of course, big data analysis. That's an opportunity. In this particular really cool study, they were correlating specific networks with lifestyle choices and able to demonstrate certain networks are very revealing of what lifestyle choices you have or how with it you are or what your vocabulary is versus not. Also, genetic correlations as well have been done. This is some nice work by Carla Miller. And I'll skip that and come to the concluding slides. Current challenges. I went over all these. And this is sort of like the big, I feel the large areas of study that might have an impact in the future. And I placed them sort of relative to where they are in this technology, methodology, interpretation, applications diagram. So noise characterization and removal, it's smack dab in the middle. It's relevant to everything. Well-curated databases, somewhere near applications and methodology. Computational models rather than description. A lot of time for fMRI data, I think we're moving in the direction of, instead of saying, oh, look, the fusiform turned on. And that's great, so let's describe that. It's like the early days of biology or botany, where you just simply describe things. And you made a nice database of descriptions. We need to go beyond that. So what we're doing now is making computational models at this level of systems to make more sense of what it means when the fusiform gyrus turns on relative to something else. And so it's a more insightful and in-depth way of looking at your fMRI data. So that needs to grow. Everything from the technology standpoint, we're still getting better MRI tricks, like things like compressed sensing, MR fingerprinting, basically cramming as much contrast into the data as much as possible. We need to still further understand the bold neural activity comparisons, neuromodulation effects on connectivity. So that's a ripe area for research. Let's see, yeah, okay, real-time feedback. So real-time fMRI is going to be talked about during this course as well. That's something that has potentially profound clinical applications, and it seems to work in many contexts. Machine learning methods, very much in methodology. It's having a big impact. We're looking more at layers. So I was going to show you some results of looking at layer-dependent activity. And if you can get at layer-dependent activity, that could also revolutionize fMRI. Because once you get at layers, there's a lot we know about input versus output in layers. And you can start to get at causality with layers. So if you know that an upper layer is active, you know that some area is acting upon it. If you see a lower layer is active, you know that it's acting upon some other area. So then you can start to make real circuit diagrams in the brain by getting layers. Okay. Multiple simultaneous contrasts. There's a lot of room in MRI to collect T1, T2, perfusion, blood volume, all at the same time. And that you can get more metabolic information. We're very much involved in the area of biomarker discovery. So we're trying to find out nice biomarkers so that we can then take a subject, put them in the scanner, and say, oh, you match or don't match this template, this massively multivariate template that we derived from this data set of like 1,000 people. And that might be a direction in which neuropsychiatric applications go. Standardized parcellation, as I mentioned, that seems like a key issue in terms of development. Local grading coils. The field is going back. Remember I showed you the local grading coil at the very beginning. Now it's going back to local grading coils just to avoid some of the DBDT issues and even push the performance even further for DTI. And individual assessment rather than group assessment. That's sort of like a holy grail of fMRI that will make an inroad into clinical applications. Okay, finally, ultimate limits and applications. Second to last slide, I promise. So what's solvable? I think we can remove all our physiologic noise. I think it's possible. Shim is also solvable as well with enough shim coils. And we can understand, I believe we can understand the completely resting state connectivity. It will take some work. We can characterize variability and actually come up with ways of characterizing at an individual subject level. We need to get control of our vendors and scanners and upgrades and pulse sequences so that we can feed into large data sets similar types of data. So that's a solvable problem. It just causes having the vendors talk to each other or coming up with some standards of acquisition, which we don't have right now. So, I mean, all of our technology, all of the really interesting things that we have, like a USB stick, that's a standard that we came up with that we agreed upon. Otherwise, we wouldn't be able to use USB sticks. It would be crazy. Same thing with fMRI. We're in that state before the USB stick of imaging. there's all kinds of variability. A universal power isolation scheme, I believe that's possible. Cryogen-free scanners, up to 70. I believe that's also possible. There's better refrigeration techniques that will bring down the scanner maintenance costs. Silent fMRI sequence, they've come out in the past and they've sort of died down because there's no clinical applications, but it's also possible to not have to ramp the gradients so rapidly, and therefore they're silent. So you can get silent ones. So what may become common practice? So sub-millimeter functional resolution for the whole brain, simultaneous multiple hemodynamic contrasts, quantitative baseline CMRO2 mapping, database research might become much more prevalent, especially it's more cost effective too. You have one massive database that's so multivariate, it's much more cost effective just to sort of mine these databases if they're well curated. Naturalistic paradigms, I believe, will be much more common as we become more efficient in characterizing what's in the paradigms and then pulling out that information. And then finally, applications. I believe we'll have, at least clinically, a lot of clinical applications, maybe for localizing function for neuromodulation or whatever, but also individual fMRI-based stress tests. So you put a subject in a scanner, you have them watch a movie, you look at their activation, compare it to what's a template of dynamics, and then you're able to say, you might have this disease in the future, or you might benefit from this medication right now. You might, the type of movie would be something that might induce anxiety, it might induce emotions of some sort, but that will be a sort of a stress test. We do this in cardiac imaging. You need a stress test. You can benefit the stress test with individual fMRI as well. Real-time fMRI feedback, I think, will be much more common. And of course, it's used for pre-surgical mapping. And you might have a massive biomarker database for screening subjects as well, for all types of neurologic and psychiatric disorders. Okay, what could change everything? There's only a few here, so I'm not going to... So room temperature superconductors. So right now, the whole search for superconducting technology is kind of an art, and people are just trying things, and it either works or doesn't work. And it seems like it's very close to possible we'll come up with superconductors that will be operating at room temperature. And not only that will change the whole electrical grid problem, but that will allow you to potentially wear MR scanners on your head. If you don't need to carry around a massive cryogen to keep them cool. So that's one thing. So if direct neural contrast if MRI is developed or discovered, that seems like a long shot. I don't know exactly how that would be, but that would change everything. So this is important. So if we have one clinical, unique and powerful and cost-effective clinical application of fMRI, that would change the game, because there's so much, like I said, standardization. There's so much work that we would benefit from if the vendors finally found it to be cost-effective to work on functional MRI. Because right now, it's like we make do. So for instance, if we have a new pulse sequence that we develop, like some brand new, like Vaso, for instance, it's not a product. There's no way of getting it propagated other than with research agreements with vendors, and then you can share maybe with one or two other people or groups. So it's not spread or tried as rapidly as they would be if the vendor cared about that. So that's a key thing, and I think that would change everything. If you had any sort of breakthroughs, I mean, there's always people coming up with function-specific contrasts that are either sensitive, maybe like labeled agents that are susceptibility agents, just like they use in PET. If you could have agents that bind to dopamine receptors that create enough of a signal change with MRI, that would be potentially game-changing as well. So in conclusion, this is actually some early work. Let's see, about 150 years ago, of Angelo Masso, one of the first people to look at brain activation by blood flow. So in some sense, he basically placed people on a balance, a very careful balance, and then he very, very delicately balanced balance, and gave them certain stimulation, had them do mathematical problems or something, And as the blood rushed more into their brain, the balance would tip, and that would show that the blood flowed into the brain. There's a whole book written about this guy and these experiments. But my feeling is that in the next 150 years, fMRI will look pretty much like this to those people 150 years from now. So we have a lot to look forward to, especially in the future and also for this course. So this is my core and groups and things like that. So I don't know, it's been long, but do you have any questions? And you can always email me questions or whatever as well. So, all right, thanks for your attention.